Hi, good morning everyone. I'm Pauline Zhang. So a bit of background information. Infections with Enterobacteriaceae can cause negative consequences for the poultry industry. A couple of examples of infections are colobacillosis and salmonellosis, and the effects can include decreased growth, decreased egg production, increased mortality, and increased carcass condemnation. Since 2014, the Canadian poultry industry has eliminated the use of extended spectrum cephalosporins, or ESCs. However, it's important to know the current situation of ESC resistance for public health. Resistant bacteria and their genes can be transferred to other animals and humans. Furthermore, currently the role of Enterobacteriaceae, other than E. coli and Salmonella, is almost unknown, so we don't know if there's transfer of resistance genes happening between E. coli and other Enterobacteriaceae. So my research hypothesis is that due to widespread horizontal gene transfer between bacteria within a host, ESC resistance genes will be similar between commensals and pathogens from chickens. So what we're really trying to look at is whether there's transfer of resistance genes occurring. So my first objective was to assess the occurrence of ESC resistance in Enterobacteriaceae, such as E. coli, Salmonella, and Proteus. My second objective was to compare the distribution of ESC resistance genes and their variants between different Enterobacteriaceae species, as well as between clinical and generic cecal and clinical isolates. My third objective was to assess the frequency of cecal carriage of ESC-resistant Enterobacteriaceae. How frequent is it that these chickens have resistant bacteria in their gut? So moving on to my methods, my sample collection was of two parts. I obtained clinical isolates from AHL that were cultured using non-selective media, and I obtained cecal samples from through CPARS from Ontario flocks. And I cultured these isolates using non-selective media. And this is in contrast to routine CPARS testing that is done with selective media. So I took these cecal samples and I incubated them so that any resistant bacteria would grow, and I plated it onto chromogenic agar. I then took all the colonies with different morphologies and confirmed their expression of ESC resistance using dis-diffusion susceptibility testing. I then identified the bacteria down to the species level using Multitoph mass spectrometry, and with both my clinical and cecal isolates, I'll screen them for major resistance gene families using PCR. So overall, for my results, I received 242 cecal samples from CPARS, and I found that 98% of them had ESC-resistant Enterobacteriaceae. In contrast to the CPARS testing that was done in 2015, they found that 16% of their E. coli were ESC-resistant. From taking all the colonies with different morphologies, I obtained 554 ESC-resistant isolates, and 90% are suspected AMP-C producers, and 8% are suspected ESBL producers. For my clinical isolates, I had 380, and I found that the overall prevalence of ESC resistance was 18% using non-selective media. Of these 67 ESC resistant isolates, 90% are suspected AMP-C producers, and 10% are suspected ESBL producers. And in comparing the prevalence of ESC resistance in E. coli at healthy chickens from slaughter for CPARS versus my clinical isolates, 16% versus 18%. The percentages are similar but slightly higher in clinical isolates. So in conclusion, the majority of my cecal and clinical isolates were E. coli. A couple others were Proteus mirabilis and Escherichia fergusoni. And the proportions of my different ESC resistance genes between my cecal and my clinical isolates were quite similar. This supports my hypothesis in that transfer of resistance genes is happening between commensals and pathogens. And finally, a large proportion of chickens carry ESC-resistant E. coli in their gut. However, in comparison to CPARS data, they found a prevalence of only 16%, and I used selective media. So this implies that it's only present in low concentrations, and that most are only detectable using selective media. And with that, I'd like to thank my advisory committee, Dr. Patrick Berlin, Dr. Georgia Slavic, and Dr. Richard Reed-Smith, members of the Berlin Lab, AHL, and CPARS, and thank you for the support from the University of Guelph, the Public Health Agency of Canada, and NSERC. Thank you.